In the session 5, you have learnt about different types of ART, that is assisted reproductive technology. Coming to the first type of artificial reproductive technology, that is assisted reproductive technology. First is artificial insemination. Artificial insemination by husband or artificial insemination by the donor. So, what is meant by artificial insemination by husband? It is a procedure in which a fine catheter or a tube is inserted through the cervix, the natural opening of the uterus, into the uterus or the womb to deposit the sperm sample. So, from the women's mate directly into the uterus. So, the purpose of this procedure is to achieve fertilization and pregnancy in women. So, the AIH is distinguished from artificial insemination by the donor, in which the donor is a man other than the woman's mate, whereas AIH is also known as homologous insemination. So, this is the what is meant by artificial insemination, one of the major assisted reproductive technology. The next is the in vitro fertilization. So, as the name itself indicates, in vitro means outside, it may be in the glass or it may be in the test tubes. So, using this IVF, that is in vitro fertilization, the eggs are removed from the mature follicles within the ovary, and the egg is mixed with the sperm in a petri dish or in a test tube. So, the fertilized, the fusion of the gametes, that is the male haploid number of chromosomes from the male and haploid number of chromosomes from the female are fused together to form a diploid zygote, and this zygote undergoes cleavage to form blastocyst stage, and that results it is transformed into the uterus of the women. So, this is what is called as in vitro fertilization, and in detail it will be explained in the further slides. Next is direct intraperitoneal insemination. So, what is meant by direct intraperitoneal insemination? It was first found out in the year 1980, which is occasionally used where the doctor is injected with a sperm into the lower abdomen through surgical hole or incision. So, with the intention of letting the sperm to find out the oocyte at the ovary after entering the genital tract through the ostium of the fallopian tube. This is what is called as DIPI, that is direct intraperitoneal insemination. This is another type of ART, that is assisted reproductive technology. Coming to the next type of ART is Gamete intrafallopian transfer, that is otherwise called as a gift. So, this is a technique in which the male and the female germ cells are required to begin the formation of a human embryo. These are injected into the human fallopian tube of the female for fertilization. And this technique is one of the methods where the doctor used to overcome the infertility problem, so the inability of the couple to produce the offsprings of their own. So, by the method of gift, the pregnancy can be achieved by the females. Next, coming to the another type of ART is the zygote intrafallopian transfer. So, this is otherwise called as GIFT, where it combines the in vitro fertilization IVF and GIFT, GIFT, that is gamete intrafallopian transfer. The eggs are stimulated and collected using the IVF technology, the IVF methods, and the eggs are mixed with the sperm in the laboratory to fertilize the egg. And results in the formation of the zygote, and then they are laparoscopically returned to the fallopian tube, where they will be carried into the uterus. So, this is what is called as GIFT. You can see in the picture how the fertilized eggs are removed, and then it is fused with the spermatozoa, and then the zygote is transplanted into the fallopian tube, where further process occur in the uterus of the women. So, this is what is called as a zygote intrafallopian transfer. Next, coming to the another type of ART is intracytoplasmic sperm injection. What is meant by intracytoplasmic? It is an in vitro fertilization a procedure in which the single sperm cell is injected directly into the cytoplasm of the egg. So the technique is used in order to prepare the gametes for the augmentation of the embryo that may be transferred to the maternal uterus. With this method, the acrosome reaction is skipped. So in this slide, in this diagram, you can see how the oocyte cytoplasm is injected with the sperm and made IV in vitro fertilization. So this is what is called as intra cytoplasmic sperm injection. Next is coming to the next type of assisted reproductive technology, that is what is called as testicular sperm extraction. It is otherwise called as TESE. It is a surgical procedure of removing a small portion of the tissues where the tissue from the testicles and where they are extracting a viable sperm from this cells from the tissues for the use of further procedure, and most commonly it is an intracytoplasmic sperm injections as a part of in vitro fertilization. And TESE is often recommended to the patient who cannot produce a sperm by ejaculation, or it may be due to the azoospermia. Azoospermia is the absence of potent spermatozoa or mature spermatozoa. Next type of in vitro fertilization or next type of assisted reproductive technology is nothing but the cryopreservation. So what is meant by cryopreservation? It is nothing but the ability to freeze and thaw the retention of viability, which provides flexibility in the human infertility therapy, where the gametes or the embryos are handled in vitro because of frozen tissues which can be stored indefinitely in the liquid nitrogen at the temperature of minus 196 degrees centigrade, and the sperm may be preserved by this technique, that is by the cryopreservation. That is what is called a sperm bank, or it may be overpreservation by the ova bank. So these are another type of technology that is to cure the infertility problem in case of male or in case of female. That is how is by cryopreservation. Next, coming to the cytoplasmic transfer, or otherwise called as ooplasmic transfer. So this is another type of ART, that is assisted reproductive technology, where the ooplasmic transfer is also a cytoplasmic transfer. It is an experimental fertility technique that involves injecting a small amount of ooplasm from the egg of the fertile woman into the egg of the woman whose fertility is compressed, and the modified egg is then fertilized with a sperm and it is implanted into the uterus of the woman who is attempting to achieve the pregnancy. So in this diagram you can see that how the cytoplasm of the donor egg and recipient egg with the recipient egg is mixed. That is originally developed in the year 1980, and the mixture of mitochondrial genetic material, and it was from 1996 to 2001. That is first experimented by Cohen, who performed this experiment, and he did it for 37 times and producing about 17 babies for 13 couples to boost the egg of the older women who wanted to conceive but were having a problem. So at this time, this technology is very important, that is cytoplasmic transfer from a potent women to the impotent women, or ooplasmic transfer. So out of 3 children born through the technique were found to have a developmental disorder. It is 2 cases of Turner syndrome and 1 cases of pervasive developmental disorder. So there may be defect also in assisted reproductive technology.